Hello everyone. Okay, hello. So we'll start today's session. So I am Shobik. I'll do integrated PhD in Indian Institute of Science, Bangalore. And today's session will be TS session 6. And in today's class, we'll discuss about some of the immunology and the vesicular, some of the vesicular transport and some of the problems. So let's start. Share the screen. Something's problem. Share screen. Yeah. Okay. So all of today's images I've taken from Alberts, and again I'll put the ebook on the drive folder so you can always access the book. So first thing is like what is immune system? So there are so immune system is the defense system of the body. So as our so as our country or any country has its own defense system to prevent any kind of external infiltration or any external infiltration, then that's like that our body also have our immune system. So our immune system is comprised of different types of cells. So all of these immune cells and blood cells actually comes from hematopoietic stem cells, and from hematopoietic stem cells there are several other cells that comes like dendritic cells, macrophage, neutrophil, eosinophil, basophil, as well as platelets and erythrocyte, which are not part of the immune system. Also, there are lymphoid cells like natural killer cells, helper T cells, cytotoxic cells, and B cells. So, as you know, in among this, dendritic cells, macrophage, as well as monocytes. So, dendritic cells as well as monocytes and macrophage, these are phagocytic cells, and these are also known as the antigen presenting cells. So and also there are other cells like T cells. So there are two types of T cells. Two types of lymphoid cells. One is B cell, other is T cell. Now, T cells can be of mainly can be of two types. So there are other T cells also, helper T cells and the cytotoxic T cells. Okay. So yeah, so T helper cells or CD4 plus T cells are the CD4 plus T cells are the CG helper cells, and these people actually help in antibody uh, yeah. So CD helper T cells or CD4 plus T cells, which are the surface marker of CD cells. These cells, T helper cells, helps in activating the immune system. However, the cytotoxic T cells or CD8 plus T cells, they helps in killing of the infected cell or killing of the infected cell or killing of the any kind of damaged cells. Okay. Whereas B cells are mainly main function of the B cell is to produce antibodies. There can be surface antibodies as well as the circulating antibodies. Okay. And yeah, so these are mainly the cells of the immune systems. And as you know, there are several immune organs of our body. These cells are generated in the bone marrow, the preliminary. Then the T cells is generated from the thymus, and the B cells is T cells move to the thymus. The progenitor of the T cells move to the thymus, and T cells is generated from there. Also, there are lymph nodes which are basically part of the lymphatic systems, and these are also known as the secondary lymphoid organs. Correct? Yeah. So yeah. So this is another image of a lymph node. Okay. And yeah. So there are mainly two types of immunity in our body. One is innate immunity, and then one is the adaptive immunity. So innate immunity, as you can understand, it's present from the birth. So it's mainly non-specific, and it protects at the first line of defense. The innate immune system includes the skin, because skin, as you can think of for a bacteria, skin is if you compare the size of the bacteria and immunes compared to a, so if this is our skins, so this acts as a barrier for the bacteria to be enter, right? So this can act as so if the bacteria wants to enter, it has to pass through this thick skin barrier. So skin itself is a part of the defense, correct? And with respect to bacteria, you can think of a wall of our house. You have to cross a thick wall of a house to go inside the house, correct? Then there are mucous membranes and their secretions, and also there are competitions with the normal microbiota. Normal microbiota means there are microbes present in our body, in our saliva, in our skin, as well as in our guts. Everywhere, if you can think of the body, most of the places there will be a normal microbiota, which has several different roles to play, including producing different metabolites. Okay. Then there is second line of defense, where comes the circulating cells. So the phagocytic white blood cells like monocytes or neutrophils or macrophages or dendritic cells, even they take the cells, so they phagocytose the cells. Okay. So during this first line of defense, it's during the second line of defense, bacteria has been phagocytosed, and there is inflammation and fever, and antimicrobial substances also has been released. Correct? And this second line of defense also include toll-like receptors, as I have discussed in one of the previous classes. Then comes the third line of defense, that is the adaptive immunity. Now, adaptive immunity, it takes some time. So innate immunity is at the real time. So there is no time lag between action of the innate immunity and the pathogen infection. As soon as the pathogen infects, the phagocytic cells as well as inflammation and fever starts. The second line of the third line of defense, or the adaptive immunity, is, or acquired immunity, is, as you can understand, it's adapts. So it has a specific resistance. That means a particular type of pathogen will have a will activate specific types of cells. So there are specialized cells like B cells and T cells, as I have discussed, and there's antibodies to make the antibodies. So this B cells makes the humoral immunity, and the T cells makes the cell-mediated immunity. Correct? Yeah. So these are the basics of our immunity, and I'll not go into very details of each of this pathway because that itself will take long time. But in this course, I think we can little bit discuss about the antigen presenting pathways. So MHC, or major histocompatibility complex. So what are MHC molecules? So MHC molecules are type of molecules which is used by the antigen presenting cells to show off the antigen on the surface of the cells on the cell surface. So if I draw it, so say this is my APC. Okay, this is one macrophage. Okay, one macrophage, and this particular macrophage will, if the pathogen enters, if a pathogen enters from outside, so I'll say this, if this is my bacteria, I'll say I make it a little bigger. So if this is my bacteria, when the bacteria enters into the macrophage, it gets phagocytosed by the macrophage, then it comes inside, and there are several processes with which I will discuss, with several process, this particular bacteria get chopped off. Okay, this bacteria gets chopped off, and then those are presented on a cell surface with help of this MHC molecule. So as you can see, so this can be of, these are the MHC molecules. Now what they will do, the macrophages, if this is a MHC molecule, it will use this MHC molecule to present the bacterial antigen. So this is the MHC and this is the bacteria, okay, or the epitope rather, or the antigen, sorry, I'll say the antigen, okay, antigen that has been come from the bacteria. So there are two classes of major histocompatibility complex. They are structurally structurally as well as functionally defined. I'm not going into structure, I'm just coming to the function, okay. So there are class 1 MHC or type 1 MHC, and there is class 2 MHC, okay. So class 1 MHC, as you should remember that for class 1 MHC, they are responsible to present the endogenous antigens like virus, mainly like virus, or the bacteria that have entered into the body. So mainly the virus is the thing that has been presented by the MHC one type of cells, and there is class 2 where there is a exogenous antigen. So sorry, the class 2 pathway is mainly responsible for the presenting the exogenous antigen, okay. And so as you can see, this is the exogenous antigen for the class 2, okay. Yeah, and class 1 MHC present antigen to the CD8 plus cytotoxic T cells, okay. Okay, and whereas the class 2 MHC, they present cells to the CD4 plus T helper cells, okay. So this is a very general phenomena. There are certain, of course, there are certain exception from this, but this is the overall, this is the pathway, okay. So now, let's go about the endogenous pathway. So as you can see, as the endogenous antigen enters, it's processed by different proteosomes, and it comes so as you can see here, so these proteins are processed by proteosomes to make small fragments. As you can see, there is a big bacteria or some bacteria, endogenous it's endogenous. It is some part of the DNA. It can be viral RNA or viral DNA, anything. Then inside the cell, there are several, inside the rough endoplazmic reticulum, there are several factors like calreticulin, ERP57, TAP. There are several other factors which helps in processing of those antigen, and now that antigen is now bind to this MHC molecule here. Now with this MHC molecule, this is further processed from Golgi, and as you can see that it has been shown as it comes and matures. That is important from the react to Golgi complex and to the plasma membrane. In case of exogenous antigen, instead of MHC class 2, which presents the endogenous antigen, the antigen is already pre-processed, and invariant chain is already bound to that processed class 2 MHC. This invariant chain helps to block the binding of endogenous antigen, because it will only bind to the exogenous antigen. Now what happens? This endogenous, so this invariant chain bound MHC class 2 complex comes to Golgi complex. In Golgi complex, this invariant chain goes off, and at some point it's circulating inside the cells. At some point it fuses with the exogenous antigens, which comes to another receptor mediated endocytosis, and these two vesicles, as you can see, they are fusing here. These two vesicles are fusing here, and the fusion of the vesicle makes such that the exogenous antigen as well as the MHC are present now in the same compartment. Now these both are sent to the they're sent to the surface of the class 2 surface of the cells to present the antigen with MHC class 2 pathway. This is a famous video where you can see that it is a bacteria and a macrophage is kind of like chasing the bacteria and it's engulfing the bacteria. This is the bacteria. Okay, I'll also once this is the bacteria here. You can see the macrophage is the red blood cells. See, it engulfed the bacteria. See once more. Yeah, it's engulfed there. This is a famous video of phagocytosis showing in the live cells. Okay. As you know, the cells important for phagocytosis is neutrophils and monocytes. And monocytes, they mature to macrophages. Okay. So this is a small video where I can show it's showing how the immune system actually functions. It's a interesting video, so I would like you to see the video. One second, I should. I should present with the sound, otherwise sound won't come.
Every second of your life, you are under attack. Billions of bacteria, viruses, and fungi are trying to make you their home. So our bodies have developed a super complex little army with guards, soldiers, intelligence, weapons factories, and communicators to protect you from, well, dying. For this video, let's assume the immune system has 12 different jobs. For example, kill enemies, communicate, etc. And it has 21 different cells and 2 protein forces. These cells have up to 4 different jobs. Let's assign them. Here are the interactions. Now, let's make this understandable. First of all, let's add colors to the jobs. Now, let's illustrate the cells. The central color represents the main job of the cell, while the surrounding ones represent secondary duties. Now the immune system looks like this. Now the interactions. Isn't this complexity just awesome? For this video, we will only talk about these cells and ignore the rest. So what happens in the case of an infection? It's a beautiful day when suddenly a wild rusty nail appears and you cut yourself. The first barrier of the immune system is breached, your skin. Nearby bacteria seize on the opportunity and enter your wound. They start using up the body's resources and double their numbers about every 20 minutes. At first, they fly under the radar, but when a certain bacteria population is reached, they change their behavior and start to damage the body by changing the environment around them. The immune system has to stop them as fast as possible. First of all, your guard cells, known as macrophages, intervene. They are huge cells that guard every border region of the body. Most of the time, they alone can suffocate an attack because they can devour up to 100 intruders each. They swallow the intruder whole and trap it inside a membrane. Then the enemy gets broken down by enzymes and is killed. On top of that, they cause inflammation by ordering the blood vessels to release water into the battlefield, so fighting becomes easier. You notice this as a very mild swelling. When the macrophages fight for too long, they call in heavy backup by releasing messenger proteins that communicate location and urgency. Neutrophils leave their patrol routes in the blood and move to the battlefield. The neutrophils fight so furiously that they kill healthy cells in the process. On top of that, they generate barriers that trap and kill the bacteria. They are indeed so deadly that they evolved to commit suicide after 5 days to prevent them from causing too much damage. If this is not enough to stop the invasion, the brain of the immune system kicks in. The dendritic cell gets active. It reacts to the signals of the soldiers and starts collecting samples from the enemies. They rip them into pieces and present the parts on their outer layer. Now, the dendritic cell makes a crucial decision. Should they call for antivirus forces that eradicate infected body cells or an army of bacteria killers? In this case, antibacteria forces are necessary. It then travels to the closest lymph node in about a day. Here, billions of helper and killer T cells are waiting to be activated. When T cells are born, they go through a difficult and complicated training process, and only a quarter survives. The surviving cells are equipped with a specific setup. And the dendritic cell is on its way looking for a helper T cell with a setup that's just right. It's looking for a helper T cell that can bind to the parts of the intruders which the dendritic cell has presented on its membrane. When it finally finds one, a chain reaction takes place. The helper T cell is activated. It quickly duplicates thousands of times. Some become memory T cells that stay in the lymph node and will make you practically immune against this enemy. Some travel to the field of battle to help out. And the third group goes on to travel to the center of the lymph node to activate a very powerful weapons factory. Like the T cells, they're born with a specific setup. And when a B cell and a T cell with the same setup meet, hell breaks loose. The B cell duplicates rapidly and starts producing millions of little weapons. They work so hard that they would literally die from exhaustion very fast. Here, helper T cells play another important role. They stimulate the hard-working factories and tell them, "Don't die yet, we still need you. Keep going." This also ensures that the factories die if the infection is over, so the body doesn't waste energy or hurt itself. But what is produced by the B cells? You've heard of them, of course, antibodies. Little proteins that are engineered to bind to the surface of the specific intruder. There are even different kinds of antibodies that have slightly different jobs. The helper T cells tell the plasma cells which type is needed the most in this particular invasion. Millions of them flood the blood and saturate the body. Meanwhile, at the site of infection, the situation is getting dire. The intruders have multiplied in number and start hurting the body. Guard and attack cells fight hard, but also die in the process. Helper T cells support them by ordering them to be more aggressive and to stay alive longer. But without help, they can't overwhelm the bacteria. But now the second line of defense arrives. Billions of antibodies flood the battlefield and disable lots of the intruders, rendering them helpless or killing them in the process. They also stun the bacteria and make them an easy target. Their back is built to connect to killer cells, so they can connect and kill the enemy more easily. Macrophages are especially good at moving up the bacteria which antibodies have attached to. Now, the balance shifts. In a team effort, the infection is wiped out. At this point, millions of body cells have already died. No big deal, the losses are quickly replenished. Most immune cells are now useless, and without the constant signals, they commit suicide, so as not to waste any resources. But some stay behind, the memory cells. If this enemy is encountered ever again in the future, they will be ready for it and probably kill it before you even notice. This was a very simplified explanation of parts of the immune system at work. Can you imagine how complex this system is? Even at this level, when we ignore so many players and all the chemistry. Life is awfully complicated, but if we take the time to understand it, we always encounter endless wonders and great beauty.
Okay, so that was about somehow this is a very simplified video. So it's a very simplified video of them, and now I'll come to a application based thing, which is flow cytometry. Now, flow cytometry can be used to in different fields. One of the major application of flow cytometry is in the immunology field, and so I'll give you a brief of how flow cytometry works. So, so because as flow cytometry has lots of things to do with I hope I have, yeah. So flow cytometry is one of the thing that also depends on something known as antigen antibody reaction. And as you see, these are antigen binding sites which have like the variable region as well as the constant region. These constant regions of the or the heavy chains are specific for a particular individual or maybe particular species. This in between the multiple in a species, this particular thing doesn't change. The variable site which is responsible for the binding to the antigens, it has can be of millions of different types. And as you can see, if there is an antigen, the antigen molecule can have different type of epitopes. So this in this epitope, the variable region or this epitope part is mainly is responsible to bind to the different epitopes on the bind board. So this particular antibody B's epitope will be different from antibody A's epitope, correct? So that's sorry, epitope is paratope. So the epitope is present in the antigen and the paratope, so this part is present on the antibody. So the variable region of a particular antibody will be specific to a particular epitope. Okay. And now there are monoclonal antibody as well as polyclonal antibody. So whenever you put an antigen in your body, the antigen will be chopped up and there will be different kind of epitopes will form, correct? And due to that, the antibody which we will generate will not be specific to a particular epitope, but it will have a, how do I say, it will have a, a league of antibodies or a group of antibodies which will act to some part of the antigen. Now for some of the research purpose we need monoclonal antibody. Now how do we isolate monoclonal antibody? I think sir has also I've explained this very well, but I'll just briefly input. So when there is an antigen, there will be like say there are four epitopes: epitope 1, epitope 2, epitope 3, and epitope 4. Correct? And these four antigens. So there are four antigens, and this image I have taken from Kubi's immunology by the way. I'll also put a link of Kubi's immunology. And so as you can see, there are so when the yeah, so when we immunize the mouse and isolate the serum from the mouse, there will be antibodies against all of this particular epitopes, correct? And so this mixture will be a polyclonal antibody. Now what we'll do is that there are myeloma cells which have the ability to indefinitely. The idea is to hybridomize that there are myeloma cells. Now, what is the problem with the B cells? The problem with B cells is that these cells die very fast. Okay, we cannot just isolate one of them and ask them to produce antibodies. What people generally do is that they use something known as hybridoma. Now in hybridoma, the idea is that there are myeloma cells or cancer cells which can be now fused with these B cells. Now these myeloma cells have a potential, which is a cancer cell, they have a potency to divide indefinitely, correct? They have potency to divide indefinitely. However, so when they fuse but they cannot produce antibody, but the B cells have a feature to produce antibodies, correct? Now if we merge these two, then the antibody producing feature as well as the of the B cells and the indefinite division feature of the cancer cells are merged. Now how to isolate? Now there will be several types of cells, right? So how do you then several, because there will be total three types of cells as previously mentioned. There will be total three types of cells. There will be H cells, correct, or hybridoma cells. Or hybridoma cells are basically what? Now B cells and myeloma cells, correct? BM. Then there would be the B cells or B cell fusion, because this fusion is we are doing it with polyethylene glycol or PEG. PEG. We use this word fusion, correct? Membrane fusion. There will be three types of BB and MM. Okay. BB cells are, they don't have a property for indefinite. They don't have a property to grow indefinitely. So these cells will apoptosize, correct? Die by apoptosis. So natural death. Okay. However, these cells are the problematic because these cells will have an indefinite tendency to grow. That's why people use a specific kind of medium or known as hat medium. What does hat medium do? It has three types of medium: hypoxanthine, aminopterin, and thymidine. Now there are three pathways of there are two pathways of nucleotide biosynthesis. One is the de novo pathway. One is the de novo pathway, and another is the salvage pathway. Okay, you can go and have a look at how these pathways work. But so what does this was? This aminopterin, this go and blocks the de novo pathway, correct? Now this MM cells or the myeloma cells, they produce biosynthesis only through the de novo pathway. They don't have the salvage pathway because of some mutation. They don't have the salvage pathway. Okay, but now since the de novo pathway is blocked by aminopterin, what is the alternative? Is the salvage pathway. So that's why these cells will die off also. Now what is here? Here in this particular cells or BM or hybridoma cells, the B cells have a proper salvage pathway. So they can use hypoxanthine and thymidine for the salvage pathway. And so they can use the B cells. These fusion cells can use a proper salvage pathway because they are fused with the B cells. Now B cells DNA will have a proper salvage pathway. Now these cells are only the antibody producing cells, correct? Now what we do? We select the cells and then we dilute it such that only one cell present in each of the test tubes, and then we further process that to further select that so that they divide a whole lot of antibody. Okay, so this is how the hybridoma technology works. So remember, there is a hypoxanthine, aminopterin, and thymidine. These are the medium, and hypoxanthine and thymidine helps in the salvage pathway. Aminopterin probably blocks the de novo pathway. Why you are blocking the de novo pathway? Because to kill the myeloma cells, correct? Okay, yeah. So actually there should be, I should also mention, there cannot be 100% fusion, right? So there should be actually total 5 types of cells. One should be only B cells, which will again die by apoptosis, and only M cells. These M cells will also because of this particular thing. Okay. What is flow cytometry? Now flow is like something is present in a stream, cyto means cells, and metry means measurements. So flow cytometry is a measurement of cellular properties like size, granularity of individual cells in a singular flow of cells. So Leonard Herzenberg from Stanford University first developed this fluorescence activated cell sorter, or FACS. Now how does flow cytometry works? Okay, I should go. Okay, so I'll just go and I should actually first come to here. So flow cytometry has 4 plus. It has fluidics, like the fluid system, which will have a sheath fluid, optics, the illumination source, the light source, electronics to control the pulse, and the software, which is not part of the instrument. So this is at the instrument, correct? So what happens in a flow cytometry? There is a single flow of cells comes through this particular tube. How the single flow is coming? So here at this part, there is something known as sheath fluid. Okay, sheath fluid. So what happens? These sheath fluids have a flow like this direction and from this direction, correct? Now because of this flow, whatever cells are coming like this, due to this flow, there is a force that is acting on the cells to make it in a single directional flow, correct? So as you can think of this, if you put say one leaf on a narrow stream, as you can see that after a narrow stream which is coming, like it's a stream and it sometimes becomes narrower. As you can think of, if you put a leaf on the stream, since the stream is moving very fast, and you see where is the narrow part is here, the leaves are getting like clotted, and there is sometimes only one leaves are actually passing. So same thing happens with the here also, correct? Yeah. So this is how the sheath fluids work, and after the sheath fluid comes, then there are light source and there are lasers. So these lasers illuminate the cells, and there are different types of detectors. So there will be one detector that's present at the forward or the 180-degree angle from the light source, which detects the forward scatter. I will come what is forward scatter and light scatter. And another detector is present at the 90-degree of the incoming light, that detects the side scatter. Also there are different fluorescent filters which will also detect the fluorescent, which will detect the cells, identify features from the cells. Okay, so now I'll come to this forward scatter. So as you can see, forward scatter is the scattered by the light that will be produced at the front of the cells. So if the light is coming through from the left, whatever is light is scattered to the 180-degree angle from the light path or through the light path, that would be the forward scatter. Whatever's the light, if the light falls on a cells, whatever light goes except this particular, like deviate from the frontal path or to the other side sideways, known as the side scatter. So you will see bigger particles produced forward scatter light than the smaller ones, and the larger cells have forward scatter signal. So forward scatter gives you information about the cell size, and side scatter because usually provides the information about granularity and complexity of the cells. So cells with a low granularity and complexity will produce less side scatter light, while the highly granular cells with a high degree of internal complexity will result a higher side scatter. So this is the basic workflow of the flow cytometry. So generally we'll use blood or cell culture or skins and make cell suspensions. Now we'll add fluorescent antibodies to this cell. As you can see, there are fluorescent molecules are tagged and they bind to the particular types of antibody or particular types of surface features which we want to stain for. Now after staining, we'll put in this as a fax machine. We'll put it here, and here the sample is given, and in the fax machine is analyzed, and we calculate the forward scatter and the side scatter. Okay, so so when a cell passes through the light beam, considering the cell has a round shape, if this is my cell, okay, and if this is my light, so as you can see, if this is my light, when the cells first coming like this, so only this part is getting scattered, right? If this is my light, only this part is getting scattered. After that, this part, then again this part. So as you can see, first only this part is scattering, then the full cell is scattering, and then only the top part is scattering. Due to that, we have a kind of like a Gaussian curve which looks like this. Okay, and so this is known as height or h, and this particular area is known as width. So we'll often calculate the FSCH versus FSCA. So A is nothing but A is equal to the area. If we integrate the width and height, so basically, layman's term, area is equal to height into width. But here in this case, since this is a Gaussian curve, we have to integrate. So basically area is basically the yeah. So from 0 to basically t, how the height and width are. Basically from here 0 to t, we are integrating the height and width. Okay, so like that. Okay, and I will come to a next video, and as you can see, FSCH and FSCA is actually used to cut out the doublets because on debris from the analysis. How it will do, it will be explained in this particular video. It's a very explanatory, self-explanatory video, and it clears you the FSC, SSC, pulse area, height and width concept very clearly. Please have a look on the video. So as you can see that these are this FSCs depend, as I told previously, FSC will depend on the cell size and SSC will depend on the granularity, correct? So this is granularity, and this is based on the cell size, correct? So now since this is these two are dependent on cell size, so as you can think of the lymphocytes, the granulocytes are the granulocytes, or the monocytes and lymphocytes are the granulocytes, right? So there are two types of white blood cells. Can based on the granularity, we can define white blood cells or about two. These are class 12 knowledge, I think, maybe class 10. Granulocytes and agranulocytes. Okay. Agranulocytes. So granulocytes are neutrophil, eosinophil, basophil. Based on that, neutrophil, basophil, and eosinophil. So eosinophil. Okay. And agranulocytes are two types: monocytes and lymphocytes. Correct. Monocytes and lymphocytes. So as you can see here, these granular cells have more granularity, so they will have very high SSCA. The size is overall not very different, but as you can see, lymphocytes are the smallest cells. So as you can see, they have very low FSC as well as very low SSC. Then comes the monocytes, and as you can see, the granulocytes or the neutrophil, eosinophil, basophil, they have a very, there you can also see a very high uh, size scatter ability. Okay, so this is how, just without any dye, but just by seeing forward scatter and side scatter, you can clearly spot out the the different types of cells just based on their granularity. And so now many times we have to, so many times we cannot just we cannot differentiate the cell types just by granularity. So what then we do? Use fluorescence. Now as you can see, say we are isolating the CD4 type, CD4 positive T cells. What will have? The cells that don't have CD4 in their say this star is my fluorescent molecule, or this fluorescently labeled antibody. Now if there are very low number of CD4 cells, only some of, or very low number of CD4 cells, or there are some non-specific binding of the antibody, as you can see here, very low signal intensity will come. As you can see here, signal intensity is near to 0, correct? Some arbitrary unit. Okay, and there are, so that means these particular types of cells are not CD4 positives. However, when there will be a CD4 plus cells, many of these antibodies will bind to the CD4 molecules on the surface, so that will increase the intensity of the signal. So as you can see, there is a shift of the intensity from, say, mean of 0.5 to mean of 2, around 4 times increase. So that means these are the same. So negative control, isotype control peak, first log decay. So these are the log cells, like scale. Okay. So if there is 0.5 means, say what, around less than 10 cells, correct? So log 10 is equal to, wherever here we have like around 100 cells, or intensity is at the 100 level. Okay. So population stained cell give a shift and increase in the fluorescent intensity to the right. Anything within the gate marked is a positive event. Okay. And yeah, so again I am just the basic instrumentation, I will not go. And yeah, so we'll move to the questions. Okay, so B cells are involved in, of course B cells are involved in humoral immunity because they produce antigens, and we have already discussed, so I am not going to have to discuss it anymore. Okay, so humoral immunity. The bond involved is joining of the heavy chain and light chain, the hinge region. So if I go to the antibody diagram, I don't know if it is there properly, but yeah. So this part, there are actually disulfide bonds which actually holds the binding chain. So these are disulfide bonds. SS bond. Okay. So SS bond, that's oops. Um, that show the that holds the chains. Okay. The antigen binding site of antibody is of course paratope. So in antigen, if this is antigen, this particular set is known as epitope. And whichever thing is binds to the epitope or the antibody binding site of this bite is known as paratope. Good. The MHC1 class involved in antigen presentation in macrophages. So what is the antigen presence in macrophages? I have discussed this in just a while ago. Someone please tell what is the MHC class involved in antigen presentation in macrophages. So it will be class 2. Okay, because macrophages do what does macrophages do? Macrophages do phagocytosis, correct? So as I have told, phagocytosis means an MHT class 2 is used for exogenous pathway, correct? So exogenous antigens, so MHT class 2 present exogenous antigens, how would come to the cells? Through phagocytosis. So MHT class 2 molecules in the macrophages are responsible for antigen presentation. A reagent used to screen clone in hybridoma technology, in monoclonal technology, HAT medium, if you remember, HAT, correct? So H is hypoxanthine, A is aminopterin, and T is thymidine. Hypoxanthine, aminopterin, thymidine, the HAT medium, and I have discussed why we use that. If there is no agglutination is observed with anti-A, anti-B antibodies, that means no A or B type antibodies, B type antigens, correct? So there is no A or B type antigens, that means there is no antigen basically. So blood group O, because it does not reacted with any of the antibodies. The agent causes fusions of spleen and myeloma, fusion of spleen and myeloma cells. It's of course big polyethylene glycols. The biochemical cell pulse marker for detecting apoptotic cells. Apoptotic cells is detected by something known as annexin. So I have not shown an image here, but there is something known as annexin PI staining. Okay. So this annexin PI staining. This is used to stain cells for apoptotic cells, and PI binds to the DNA, and annexin binds to something known as phosphatidylserine. So this is the correct answer. Phosphatidylserine. So annexin binds to this particular thing. Okay. Which of the following application is not possible in isolating green fluorescent protein from plants? Of course, we can do. You can use fluorescence because we have fluorescent marker. To identify various types of cell types in certain diseases. Yes, we can do. We can always check if there is a particular antigen is present, and we can have an antibody against that particular antigen, and we can tag that antibody with fluorescent protein. So always we can use to find the cell types unique to certain diseases. In immunophenotyping, yes, immunophenotyping is a, I have not discussed, but immunophenotyping is a very well-known application of uh, facts. In determining amino acid sequence of in a protein, no, we cannot. This is not a sequencing technique, so we cannot. The channel attributed to granularity of the cells in the flow cytometry, as I have discussed, it's forward scatters. Forward scatters measures the granularity. Granularity. So forward scatter measures the size, and the side scatter measured the granularity. There is nothing known as upward scatter or downward scatter. The I used to analyze cell cycle in as I've told, propydium iodide just before I have asked. So what will happen in propydium iodide? This also I should explain a bit of you. Now, as you can think, during the G1 phase, the inside the cells, so propydium binds to only dsDNA. Okay, only double-stranded DNA or dsDNA. Only binds to a dsDNA. Now inside at the G1 phase, how the chromosome looks like this, correct? The chromosome looks like this, or I should not actually, no, not like this, exactly not like this. So the chromosome looks like this because there is not yet division happening, correct? So at G1 phase, now at the S phase, in S phase, DNA replication happens. So there will be no single-stranded DNA. There will be multiple single-stranded DNA, right? So in S phase, the DNA is single-stranded. Then G1, it's double-stranded. At G2M, again DNA is double-stranded, correct? But at the G2M phase, there are two double-stranded things. So one will have like this, another will also have like this, correct? So what happens if this I just say number or intensity, and this is, or I should not say number, I should rather say PI, propydium iodide intensity, correct? PI intensity. At the G1S phase, since there are all double-stranded DNA, we have a peak like this, correct? But at S phase, there will be a decrease in the peak, and again at the M phase, we'll see a peak. So, like that, then we can divide. This is the G1 phase, the S phase, and this is the G2 by M phase, correct? So like that we can see. So we will see two peaks. One is the G1 peak, another is the G2M peak. Okay. So this is how the propydium iodide strain is done. Okay. So this comes to end of our immunology part, and some of the apoptosis part and all. I'll have just 1 minute of break to have some water, and then we can start again. Okay, we'll restart again, and yeah, sorry. So in vesicular transport, it's made of like different type of secretory as well as endocytic pathways of protein sorting. Okay, so there can be two types of, mainly three types of events happening inside this vesicular transport. One is uh, endocytosis. Um, just a second. One is endocytosis, or this one, the endocytosis. Then two is exocytosis. Exocytosis, and third one is protein targeting. Correct? Protein targeting or protein sorting, whatever you do. Protein sorting. This happens internally. So, or rather, I should say intracellular transport. Intracellular cellular transport. Okay. Intracellular transport. So now, as you can see, this is the endocytic pathway. So in the endocytosis, what happens? The molecules from outside is secreted in the endocytosis. It's secreted. It's endocytosed by either receptor mediated, most time receptor mediated endocytosis, and then it forms endocytic vesicles which go and fuse into most of the time to the layer from the early endosome, late endosome, and fuse with the lysosomes, correct? Then there are different types of secretory pathways, what is known as secretory vesicles, as well as the regular two types of secretion as or exocytosis, that is regulated secretion as well as constitutive secretion. There are two types of those, but overall these are same. And then the third, intracellular sorting, like for example transport of things from Golgi to lysosome or things from Golgi to mitochondria, like that. Okay, so there are intracellular transport from to the nucleus, several things, right? So intracellular transport, there is transport between the compartments. So during this protein sorting, the protein is get synthesized, and some of the proteins are in the ER lumen, and they are matured, go to the Golgi network. From Golgi network, some of the proteins are retrieved back, and this is maturation and goes for exocytosis or the surface expression, correct? So there are different kinds of codes for doing this kind of work. Number one is the clustering codes. So clustering codes is used for endocytosis. As you can see here, the clustering coated pits are forming and it's getting endocytosed. Next is the COP1 and COP2. So COP1 is used to from, so COP1 is used from, so, sorry, COP2 rather I should first say. So COP2 is used from ER to Golgi, and COP1 vesicles are used for retrograde transport or Golgi to ER, correct? And we will go into details of this, okay, a little bit. Sure. So while for now, how does the proteins go into the ER lumen? So something that is known as the co-translation and transport of protein, or the signal hypothesis. How does it work? So if this is the ribosome, as you can see, there is something known as signal peptide and the signal receptor. So the signal receptor goes and binds to this signal peptides. So signal receptors, SRP, this goes and binds to the signal receptor, which is just came out from the ribosomal tunnel of the nascent protein. This goes and binds to the signal receptor molecules and cause a translational arrest. Now this signal receptor molecule has a the SRP. This SRP has a receptor on the ER membrane. As you can see, this is the ER lumen, this is a cytosol. So is this SRP goes and binds to this particular signal receptor, and then it has a translocator which translocates this particular signal receptor. After that, there will be a cleavage of this signal peptide, and the protein and the protein synthesis will start, and the protein is go inside the ER lumen. There are several types of signal proteins. There can be the ER transmembrane lumen proteins, or something like those are chaperones, mainly the membrane bound chaperones. How they go into? So as you can see, they have an N-terminal signal sequence. They come and they have a hydrophobic transfer sequence. So as soon as it has the as soon as this membrane starts, since this is hydrophobic, as you remember, hydrophobic regions goes and binds. So this signal peptide is cut off this signal peptide, and as you remember previously, the hydrophobic stretches don't like to be in the open lumen. So they always try to go and fit in the membrane. So this hydrophobic sequence is also known as the stops transfer sequence, because it stops the transfer of the protein inside the ER lumen. So as you can think of that, so this is the protein, this is a signal peptide, and after the signal peptide is removed, this particular it's a transmembrane protein. So this is the outer membrane cytosolic region, and this is the ER lumen region. Okay. So these proteins are either stays in the ER as a ER resident membrane protein, or it goes and binds. It takes off as a vesicle and goes and binds to the as a surface receptor protein. For example, the MHC proteins, correct, because they will have this kind of structures. So there are from ER to from the ER with the help of this COP2 code, they go and bind. Now the ER resident proteins have a particular thing is known as KDIL. So what does KDIL do? ER resident proteins bind to the yeah. So now what happens is that during the transport, the COP2 vesicle proteins or the KDIL proteins will go and okay, sorry, I'm sorry. So during the transport or the forward pathway, the ER, some proteins from the ER membrane with help of COP2 coating goes and fuses to the Golgi bodies, correct? This is the Golgi. Now what happens, some of these proteins or known as the ER proteins. So the ER resident proteins have something known as something specific is known as KDIL. So KDIL is a marker for ER resident proteins. ER resident proteins. Now you can think of something like this. Okay, now you can think of something like this. So for example, you are traveling somewhere with the aeroplane, correct? Now there are passengers as well as there are staffs. Now when the aeroplane goes and lands, you are going from say India to Europe, okay? So think of the India is your endoplasmic reticulum and the Europe is your Golgi bodies. Now the staffs who are driving the plane or the air hostesses, they go, the cargo or the persons is moved there, and with the returning flight, the drivers or the air hostesses, they come back, right? Similar happens within the cells. As you can think, there are some ER resident proteins like this KDIL proteins. Now during some of this kind of so during some transport, some of these ER resident proteins also go there to the Golgi bodies during this transport. So now they in Golgi bodies in the membrane you have something known as KDIL receptors. So these KDIL receptors bind to these KDIL proteins and using the COP1 code, they again retrieved back from the Golgis to the ER. So that is known as the retrieval pathway. So forward pathway is done by the COP2 coated vesicles. The retrieval pathway is done by the COP1 coated vesicles. With the KDIL receptors is binding to the KDIL receptors is binding to have the KDIL marker, which is the marker for the ER resident proteins. So this is a very conserved pathway. Also, you should remember this KDIL and COP2 and COP1. Then we have talked about N-linked and O-linked glycosylation, and this glycosylation itself is a complex thing. So partial glycosylation is done at the ER lumen. This type of glycosylation helps in binding of the chaperones, which helps in the folding of the protein. Then another further processing of this glycosylation is done in the Golgi lumen, correct? And there are two types of glycosylation. One is the N-linked glycosylation, which use the N group of asparagine to bind to the different kinds of oligosaccharides, or we have the O-linked glycosylation, where the serine or OH group of the serine or threonine is bind to the oligosaccharide group, correct? So in the ER, glycosylation is done by something known as the ER mediated glycosylation. In this case, basically, the whole oligosaccharide chain or this particular thing, the 3 glucose, 9 mannose, and NS2 glucosamine is synthesized, which is then directly transferred with oligosaccharide transferred to a asparagine group of the in the ER. Okay, how this particular thing is synthesized? So this is synthesized on a base of a lipid known as dolicol. So first dolicol is phosphorylated with CTP and ECTP and made the dolicol phosphate. In dolicol phosphate, the UDP first UDP N-acetyl glucosamine or 2 molecule of N-acetyl glucosamine comes and binds, which makes first the NS2 of N-acetyl glucosamine. After that, 5 mannose GDP mannose comes, and from this particular group of mannose 5, then something is an enzyme known as flippase. I should flame. So the enzyme is which is known as flippase. Okay, this flippase I don't fine. Yeah, this flipped across the bilayer. Okay, so this flippase flips this dolicol phosphate molecule from the cytosol side to the lumen side. At that position, that side also, 4 mannose group as well as 3 glucose group added. Now this whole bunch of things is now taken except the dolicol phosphates. This particular part is cleaved, and this thing is now attached to the through a N-linked glycosylation to the asparagine group to the of a protein. So this is now the N-linked glycosylation which happens in the ER lumen. So the dolicol phosphate here is acts as the base for this whole assembly. Okay, and then there are several ATP pumps also. I'm just this has not been taught, but there is a question regarding ATP pumps. So there are 4 type of pumps: P-type pumps, ABC transporters, V-type pumps, and F-type pumps. So V-type pump are proton pumps, and F-type pump are ATP synthesis pumps. So the P-type pumps, as you can think of like from the P, because they phosphorylated themselves during the pumping cycle. So they use the energy from ATP to ADP and get phosphorylated and use that to pump ions out of the cytosol. So for example, the sodium potassium Na plus K plus that pump, those are P-type pumps. Okay. Then there are ABC transporters. These transporters used to pump small molecules from across the cell membranes. These all ABC type pumps also are used to pump drugs to outside the cells. Okay. Then there are V-type pumps. So V-type pumps are proton pumps. So they using the energy of ATP, they pump H plus molecules from the cytosol to inside a particular membrane or particular organ or particular vesicle. So these is used to acidify the endosomes. So endosomes have lots of this. Endosomes have lots of this V-type proton pumps, which helps the make the as the at the endosome matures from early endosome to late endosome to the lysosome. The pH of the vesicle decreases. This is because they have lots of V times ion and they is constantly pumping H plus ion from the cytosol to the to the inside the vesicle. The opposite of the V-type pump is the F-type pump. So these pumps are found in the mitochondria or the mitochondria. So these types of pumps are found in the mitochondria, which is also known as the ATP synthesis. ATP synthase, because they pump H plus ion and H plus ion from the lumen to the cytosol, and that produces the one of 1 ATP. That produces from ADP to PI to ADP. Okay, and it's found in the plasma membrane. So ATPs are found in the plasma membrane of bacteria, the inner membrane of mitochondria, the thylakoid membrane of the chloroplast. Correct. And so during the oxidative phosphorylation in anaerobic bacteria or mitochondria, and during photosynthesis. Okay, so let's see the class of pumps used to maintain acidic pH of the lysosome. I just now discussed it's a V type pump, V class pumps. Correct. The organelle adds proteins to the glycosaminoglycans to form proteoglycans is Golgi complex, because lysosome proteins are degraded. SCR and RER, there is N-linked glycosylations, but here I am taking glycosaminoglycans to form proteoglycans. This is done in the Golgi. The sorting of newly synthesized proteins to their respective cellular destination is called protein export, import, targeting, or delivery. Of course, targeting, because a protein is newly synthesized. Correct. Now it has to go through nucleus or ER or cytosol. It doesn't. It's in the cytosol or in the mitochondria. That will depend. That will depend. So that will depend on the export or the sorting signal. The sorting signal is also known as protein targeting, because it's targeting to a particular organelle. Where does the ribosome and the polypeptide chain gets targeted once a short fragment of protein chain is synthesized? Yeah. So this short fragment here is the signal peptide, and as this is also known as the signal peptide. The signal peptide binds to the signal receptor proteins SRP, and this SRP goes to the bind to the SRP receptor SRPR in ER membrane, which is then, so the protein gets targeted to the endoplasmic reticulum. Okay, the binding of signal receptor particle or SRP, this is the signal receptor particle or SRP, this is also known as SRP, signal receptor particle, is a larger to the larger subunit during translocation leads to which of the following? As I have told you, it's a translation arrest, because translation is arrested, and after the SRP binds to the SRP receptor in the air membrane, then again the translation then starts. Protein glycosylation, glycation is always carbohydrate. Correct. The lipid that transfers the dolicol. You remember the dolicol is present in the ER glycosylation. Correct. ER glycosylation. And sugar from cytosolic side to endoplasmic side. So it's flips. Flips is done by flippase. Okay. Floppase is also another kind of. So floppase and another kind of enzyme, but in this case of flippase is the correct answer. The transfer of proteins from endoplasmic reticulum to the Golgi. So this is the not the retrograde pathway. This is the forward pathway. So forward pathway is done by COP2. The retrograde pathway is done by COP1. So correct answer is COP2. The cellular organelle involved in detoxification of the liver is smooth. It's smooth endoplasmic reticulum. So that's all for today, and Yeah. So if do you have any questions about till now, if in the class, like to know? So if there is no more questions, I'll end the session here. Thank you, everyone.